Hello, yeah, in this video, I want to show you how to perform variable clustering using the R function varclus in the Hmisc package. Okay. And as you know, the Hmisc package is written by Professor Frank Harrio. It's a very useful package, contains many, many useful R functions. So I definitely recommend you check it out. Okay. All right, so let's load the library, set random seed, um, assign a sample size, and then create 3 groups of data. And then we introduce correlations within the groups. Okay, so we get 3 groups of correlated variables. Put them into a data frame, assign column names, and this is the data look like. Okay, so this is a data structure. Okay. And to actually use the varclus function, this is one way to use it. So we need to specify the data to be entered as a data matrix. So we use s.matrix of data. And then similarity equal to Pearson. So similarity, you can have different choices of similarity measures. I recommend you look at the R documentation to get familiar with other similarity measures. Okay, so doing that, it's pretty quick doing the clustering. You plot it out, it looked like 3 clusters make more sense, right? And also you can print out the object VC, but if you wondering how do I know which variable belongs to which cluster, right? So for doing that, we need to specify the number of clusters from the graph. It looked like 3 clusters make sense, so we can use cut tree of the VC of each cluster and k equal to number of clusters. You get the membership, you print it out, it looked like that. Okay, tells you which variable belong to which cluster. Okay. I just want to take a look at the structure of this VC object and let you see the similarity and the merge, the height, and the order. Okay. The similarity is the pairwise similarity between variables, and merge is the matrix showing how clusters of variables are merged at each step. And height is the numeric vector of dissimilarities at which a merge occurred. And order is the vector specifying the x-axis order of variables in the dendrogram. So the merge, the height, the order, they're all presented in the dendrogram on the right. Okay. Oh, another thing I want to mention is there's another way we can use varclus using formula. And this has the advantage, if you want to use all the variables or you want just a few selected variables, then you can tell R using a formula. So here we're going to do all variables. So here the dot and data equal to data, similar to the Pearson. And then we can plot this out. We get the same dendrogram. The printed out is the same object output. If you really want to have a closer look at the similarity, we can extract it, and then we can say generate a heat map to visualize the correlation. Okay. So we have used the varclus on a simulated data set, but I was wondering if we can try a somewhat real data set. So I load in the golub cancer gene expression data. It's a 72 by 7,129 columns, dimension names. Okay, so let's expand the maximum number of print. And this is the, this is the all the variable names, their genes. And then I standardize this data matrix, and then it does require it input a data frame in there. Okay, so, and then I'm going to run this, and now I'll pause the video and I'll come back when the results are available. Okay. Okay, we're back. It took about a couple minutes, for R to finish this variable clustering, which is not too bad. But I remember we have over 7,000 variables, so it's really not very helpful if we want to look at the like a dendrogram. I'll try. Hope it doesn't crash my computer. Yeah, it looked like really messy. We cannot make any sense of it. But if you ask how do I know the memberships, okay? If you still remember in our last video when we used our package varclus, we used the function mlcc.bic to determine the optimal number of clusters, and it took like 24 hours for it to complete that computation. And it showed the optimal number of clusters based on BLIC is 11. So if we assign number of clusters 11 and use the cut tree function, and then we can print out the membership. So it's there's several thousand genes here, so we cannot see the whole thing, but you get an idea. So each gene belongs to one cluster. Okay, so you need to do some post hoc coding and to really make good arrangement of this membership if you need. But anyway, it looked like the R function varclus is very easy to use. I think it's built on Hclust, the function, so it's doing hierarchical cluster, and we can do additional coding to extract the membership, and it does work well on large number of correlated variables. Okay, that's a good information. Thank you.